Hi, everyone. My name is Brandon. I am a graduate student in Paul Boutros' lab. Fun fact, I was also an undergraduate student in Dr. Young's lab, so very cool to see all the work that's going on right now in the lab, and great to see how far the lab has come. All right, so the title of my talk today is Exercise and Tumor Genomic Landscapes in 2,700 patients with cancer. Here's my outline, just some background, hypothesis, the work that I'm doing, and then future directions. All right, so before I move on, let me first define exercise as physical activity that is planned, structured, repetitive with the intent of improvement or maintenance of physical fitness. So exercise is quantified in METs or metabolic equivalents. The MET is essentially a ratio of how much energy an individual is consuming relative to their baseline metabolic level. So in other words, you and I might have different baseline metabolic rates, but we are all consuming one MET right now by just sitting in front of our computers. So it's just normalized to your own metabolic rate. So we express exercise in MET hours per week, and to calculate that, you just multiply these MET values that you see on the screen for each activity times the number of hours you do that activity per week. So whenever I present about exercise oncology, people ask some sort of variation of how much exercise do I need to do, or am I doing enough exercise? So to do a fun little exercise, pun intended, I'm going to quantify my typical week of exercise, and then you can use me as a point of reference. So I just dance for 2 hours a week, I play tennis for 2 hours a week, and I go to the gym for 2 hours a week. And so after multiplying the MET values together, that puts me at about 30 MET hours a week. And so funny story, when I was presenting this slide to my thesis committee, they were like, wow, you're not spending enough time in the lab. And I was like, oh, okay. But anyways, so as we all know, cancer is one of the leading causes of death worldwide. There are many different factors that influence whether an individual is more susceptible to develop cancer, such as their genetics, their environmental exposures, and lifestyle factors. So approximately 2/3 of cancers are preventable, with some of the greatest modifiable lifestyle factors being smoking, diet, and exercise. Smoking is by far the greatest preventable cause of cancer. And in contrast, exercise is the greatest positive modifiable factor. And let me show you some evidence of that. So here is a plot of the hazard ratios of 26 different cancer types. A hazard ratio less than 1 indicates less risk of cancer with physical activity, and greater than 1 means higher risk of cancer with physical activity. And the red arrows over here just indicate 13 different types of cancer that were found to have statistically significant decreased risk with physical activity. Physical activity after cancer treatment may also improve clinical outcomes. So on the left, we have a cumulative incidence curve of colorectal cancer specific deaths by level of post-diagnosis physical activity. And on the right, we have a Kaplan-Meier curve of the overall survival. And as indicated by these red arrows, basically the most active group had the lower mortality, both cancer specific and overall. Here we're just looking at breast cancer. Once again, the Kaplan-Meier survival curve shows that the most active group has the lowest mortality. And in the table to the right, we see that same pattern with cancer recurrence. Okay. And then so hopefully by now I've convinced you that exercise is the strongest positive modifiable anti-tumorigenic factor, and that exercise can be beneficial at different stages of disease progression. So before cancer diagnosis, exercise can lower an individual's risk for cancer. During cancer treatment, exercise can help patients deal with the side effects of treatment. And then after cancer treatment, exercise may lead to better clinical outcomes. So this is important because exercise has a lot of public health potential for cancer prevention and treatment because it can be safe, accessible, and cost-effective. And also, when a patient gets diagnosed with cancer, they often ask questions like, how bad is it? How much time do I have left? What can I do to get better? And exercise has that ability to give people the agency to positively influence their own risk of cancer or cancer-related outcomes. So if anyone ever asks you, what are some ways that they can avoid getting cancer, the two simple solutions or two simple answers are really quit smoking and exercise more. So while exercise is the strongest positive modifiable risk factor, surprisingly, the molecular mechanisms by which it influences cancer is still almost entirely unknown. So basically, the problem with all the data that I've shown you is that all of those studies were observational, and there's just no understanding of the molecular mechanisms by which exercise exerts its effect on cancer. So that leads me to my project and my hypothesis, which is that exercise exerts dose and tumor-dependent effects on the clinical and molecular behaviors of the host and the tumor. And this brings me to my work. So the cohort that I am working with is from the MSK Impact Study. Basically, MSK, or the Memorial Sloan Kettering, is a big cancer center in New York that decided to create the first FDA-approved targeted tumor sequencing panel of over 500 genes, and they've used that to sequence over 65,000 patients to date. So what my collaborator, Dr. Lee Jones, did was send out exercise surveys, the IPAC, to all of those patients and asked about their post-diagnosis exercise level. And that's one source of our exercise data. Another source is the GLTech exercise survey, which is another post-diagnosis survey that patients fill out as part of their routine intake forms when they go to a cancer survivorship clinic for their follow-up appointments. And so after some quality control, this gives us a cohort of about 2,700 patients with both genomic profiling and exercise data. And to our knowledge, this is the only data set out there that has linked exercise and tumor genomics data. All right, so on the left, we have the cancer type distribution. I do a pan-cancer analysis and a cancer type specific analysis for a lot of my downstream analyses. So for the pan-cancer analysis, we combined all cancer types with a sample size greater than 10. And then for cancer type specific analyses, we only looked at cancer types with a sample size of greater than 50, which are the ones shown in this figure right here. On the right, we have the exercise distribution. Again, we quantify exercise in terms of METs. And you'll see that a little over 30% of our cohort has 0 MET hours per week, which we will use to define as non-exercisers. And any patients with greater than 0 METs will be considered an exerciser. And we will use the exerciser and non-exerciser classifications to answer questions about whether or not exercise has an effect on genomics. So yes or no, does exercise have an effect? But we'll also use the MET values themselves to answer questions about how much exercise has an effect on genomics. So does exercising more have more of an effect? So a dose response. So let's start with some univariate analyses. Here is a Spearman correlation matrix between a bunch of different numeric variables in my data set. Already you can see that exercise or METs is anti-correlated with age and BMI, which is not super surprising because, older and larger people tend to exercise less. Some of the genomic features that I'm going to be looking at are TMB, or tumor mutation burden, FGA, which is the fraction of the genome altered, and microsatellite instability. All right, so let's also take a look at individual cancer types. These are dot maps. So the size of each dot is the effect size. The color of the dot is the direction of the effect size. And then the background shading indicates the p-value. So on the left, we're looking at Spearman's correlation again, where the effect size is Spearman's rho. And on the right, we are looking at a simple linear regression where the effect size is the R squared. So again, we see that exercise is anti-correlated with age and BMI, which are these two columns right here, for most cancer types. Alcohol, smoking, and treatment are also associated with exercise in some, but not all cancer types. But ultimately, these all warrant consideration to be covariates to exercise when we model the relationship between genomic events and exercise. So those were demographic covariates. Let's look at the correlation between exercise and genomic features. This shows that exercise is associated with some genomic features in different cancer types. And long story short, I've done a bunch of other univariate analyses, but I won't bore you with the rest of it. So let's just hop on ahead to looking at genomic landscapes. So given those univariate analyses and our background knowledge, we fit this logistic regression model, mutation status as a function of exercise, adjusted for age, sex, the survey taken, their treatment status, smoking status, and alcohol status. For the pan-cancer analysis, which is the one on the left, we also adjusted for cancer type as well. So along the top, so each column is a patient, and along the top, we have patients ordered by METs. So the greatest exercisers are on the left, and as you go to the right, they're exercising less. Recall that in my little example, I had around 30 MET hours a week, so I kind of fit in the upper 1/3 of these patients. So each row is a gene with mutations, and the color indicates what type of mutation is present. So lastly, the p-values are in this bar graph to the right. And so, for example, here, CDKN2B has more mutations among patients with less exercise, and these mutations tend to be CNAs, which are purple. And then also ERBB2 tends to have more mutations among higher exercisers with a variety of mutation types. You see like purple and yellow, which are the single nucleotide variants. And then so looking at melanoma, again, we just see more mutations amongst higher exercisers in BRAF, and then more mutations among lower exercisers in like FAT1, for example. And these tend to be, single nucleotide variations, et cetera, et cetera. I have these for every cancer type. So next, instead of looking at exercise dose, we also looked at exercise status. On the left, we calculated the risk ratios for exercisers versus non-exercisers. So the genes with risk ratios on the right of 0 means that the exercisers have a higher risk of mutations in that gene. And then on the right over here, this bar plot just shows the percentage of patients within each group that have mutations in each gene. So for CDKN2A, about 12% of non-exercisers had mutations, and about 9% of exercisers had mutations. All right, so that was for pan-cancer. I also have this for a bunch of different cancer types. And basically the conclusion is that between the exercise dose and the exercise status analyses, we have some overlapping hits, but there are also some genes that are only hits by either looking at dose or either looking at status. This is the last piece of data I'll show today. This is a dot map showing the risk ratio for the cosmic mutation signatures. So the risk ratio is evaluating the number of exercisers within each cancer type that have had a call for one of these mutational signatures. So for example, in endometrial cancer, exercise had a higher risk ratio. for SBS5, while in prostate cancer, exercise had a lower risk ratio for SBS5. SBS5 is an unknown mutational signature. And then we have SBS13 over here, which is a signature for the activity of APOBEC. And exercisers have a lower risk of this mutational signature in both my pan-cancer analysis and in breast cancer. And so interestingly, APOBEC is commonly thought of as like a DNA mutator or RNA editor. So there seems to be a downregulation of APOBEC-related mutations in the pan-cancer and breast cancers in relation to exercise. Future directions. So lots of ongoing work. So it's great that we are finding some genomic associations with exercise, but what does this truly mean and why does it matter? So I'm an MD PhD student, so oftentimes I like to think about like, what is the clinical impact of these things? And so I want to evaluate whether these genomic associations, such as the increase or decrease of tumor mutation burden and fraction of genome altered with exercise, have any clinical impact. And I'm currently looking at mortality data in relation to TMB and FGA in some independent cohorts, both TCGA and an independent cohort that my collaborator has. And at the beginning of the talk, I showed some studies about cancer risk and mortality, but no one really has looked at the timing of cancer diagnosis. That is, like, for a patient who is already going to get cancer, does exercising prolong or shorten their time to diagnosis? And lastly, we can stratify patients into cancer groups given their polygenic risk scores and like presence of rare variants. But we can also ask the question of whether exercise can actually, for example, lower the risk of individuals who are already at a high risk for cancer given their PRSs and prevalence of rare variants. So lots of fun things to continue to study. These are my acknowledgements. Again, I'm in Paul Boutros' lab. This is a highly collaborative project with Lee Jones from MSK. And yeah, thank you for listening, and I'm happy to take questions.